Hello everyone, welcome back to my channel. Today we are going to start with another topic of developmental biology, that is gametogenesis. Let's begin. As we have discussed earlier, the phases of development in the developmental biology, and the very first step in the phases of development which we have deal with, that is gametogenesis. Today we are going to deal with only spermatogenesis. As you know, what is gametogenesis? Gametogenesis is nothing but the formation of gametes, that is male gamete and female gamete, and the formation of male gamete is known as spermatogenesis. In today's video we are going to discuss about spermatogenesis in detail. So let's begin. Before that, let's understand what is spermatogenesis. Spermatogenesis is the process in which haploid spermatozoa is developed. From where does this haploid spermatozoa is formed? From the germ cells. Where is the germ cells present? The germ cells is present in the testis, that is a male reproductive organ. Okay, the male reproductive organ consists of testis, and in testis there is seminiferous tubule, and inside the seminiferous tubule the germ cells are present which develops the haploid spermatozoa. What is haploid? Haploid means n number of chromosomes is known as haploid. Okay, so this spermatogenesis starts with the mitotic division. So let's see what is the mitotic division. As you can see in this picture, this is the primordial germ cell. What is primordial germ cell? Primordial germ cell is nothing but primitive germ cell from which the spermatogenesis process begins. Okay, and one more thing is that the spermatogenesis process is again of two types. Okay, one is spermatogenesis in which spermatozoa is formed. Another one is spermiogenesis from which the spermatozoa will form into the active or motile sperm. These are the two separate processes. First we are going to deal with the spermatogenesis. Okay, from where we get the 4 haploid spermatozoa. And this spermatogenesis process is divided into 3 phases. First is the division phase, or you can say the division phase, then is the maturation phase, and third is the growth phase. Okay, division phase, maturation phase, and the growth phase. And you can see by this that in the division phase what happens, the 2n number of chromosome undergoes the process of mitosis and forms 2 set of 2n number of chromosome, which further undergoes mitosis and form 4 2n number of chromosome, that is known as spermatogonia. Then these spermatogonia will undergo the growth phase and forms the primary spermatocyte, and from here the meiosis process starts. Till here the mitotic process was taking place. From here the meiosis process takes place. So you can say, so spermatogenesis is the mixture of both mitotic and meiosis process, not only mitosis but meiosis as well. From here the meiotic division start, and as you know, in the cell division or in the cell division chapter you might be knowing that the meiotic division is a reductional division, whereas mitotic division is a equational division. As you can see here also, that 2n number of chromosome, when it was dividing into 2 daughter cell, it is forming 2n only, no reduction. But here the primary spermatocyte will undergo meiotic division and will form n number of chromosome, that is reductional division. Further this will form again a secondary meiotic division and form 4 spermatids or 4 spermatozoa. These 4 spermatids will undergo differentiation and form spermatozoa. But here the spermatozoa which are formed are not motile. They are non-motile in condition. For motile condition they have to undergo another process, that is known as spermiogenesis. So here in detail I have mentioned what all process takes place. The mitotic division produces two types of cell, A and B, as you have seen here, A and B. Okay, then this differentiate into primary spermatocyte, and primary spermatocyte will differentiate in mitotically and form spermatid. Okay, and these develop into mature spermatozoa known as sperm cells. Okay, the spermatozoa are the mature male gametes in mainly sexually reproducing organisms. So this process of formation of spermatozoa is known as spermatogenesis. And in mammals, the spermatogenesis takes place in the testis, as I have already discussed. Okay, and this, when does this phase start? This start from the very beginning, that is from the puberty. Okay, till the time of death, the process of spermatogenesis takes place. From the puberty it starts, and then till the death it takes place. Okay, and most important thing, what are the changes which takes place during the spermatogenesis? That is, DNA methylation and histone modification takes place during the spermatogenesis. Okay, next we have seen the stages of the spermatogenesis, that is what all happens in this process, and this is a table representing the number, cell type and the number of DNA copy as well as the process of cell. That is, you can see that first is the spermatogonium, or in the primary spermatocyte diploid 2n number of chromosome, that is 46 chromosome present, and number of DNA is also 46, that is 2C. And the process is known as spermatocytogenesis. Okay, and the process, let me zoom it so that it will be clearly visible. Okay, spermatocytogenesis. And next step is when the primary spermatocyte is formed. Here also chromosome number is 46, and number of, here it is to be noted down that here the number of DNA copy is increased, that is into 4C, that is 2 into 46. Spermatogenesis, that is meiosis 1 phase. Here, from here, that is from here this stage, that is from the primary till here, the number of chromosome as well as the number of DNA copy will be same. From here, when the primary spermatocyte will be formed, that is when first meiotic division will take place, the number of chromosome will be same, but the number of DNA copy will increase. This is very, very important point. Okay, this is meiosis 1. Next come to the meiosis 2. Here in the meiosis 2, the haploid number of chromosome will form. Here you can see here the meiotic 1, 2n number of chromosome was there, but when the meiotic division 2 was taking place, number of chromosome was reduced into n, that is haploid. So here also the secondary spermatocyte, the number of chromosome was reduced to half. From 46 it became 23, but the number of chromosome became 2 into 23, 2C. This is second meiosis stage. And during spermiogenesis, that is, we have not discussed the process. During spermiogenesis, same haploid number of chromosome and DNA copy is 23, as well as in the spermatoids also 23, and chromosome, that is DNA number, also 23. So major change takes place in this stage only, that is during the meiosis 1. Rest is easy to remember. This table is very important because this table is regarding this table many questions are asked. The number of chromosome will be asked, or number of DNA copy will asked, and the processes will be asked. So this table is very important to be noted down. Okay. Next, coming to the main phase, that is spermiogenesis. This phase, that is during the spermatogenesis process, as you have seen here, wait a minute. Okay, here the number of chromosome which was formed was, that is the number of spermatozoa was 4. Okay, 4 haploid number of chromosome spermatozoa was formed, but they were all non-motile. So during this spermiogenesis process, the final stage, which is the spermatid, will develop into mature spermatozoa, or you can say sperm. Okay, and these phases again divided into 4 phases. First is the Golgi phase, then is the acrosome phase, and then formation of tail, maturation phase and spermation. Okay, so what all happens in this phase? Basically the chromosome contain the spermatozoa, nucleus, Golgi apparatus, centriole and mitochondria will be formed. Head, middle piece and tail will be formed in this process. Okay, so what happened in the Golgi phase? During this phase, the Golgi apparatus will create enzyme that will become the acrosome. And what is acrosome? Acrosome is a main part of the head of the sperm, which helps in the diffusion with that of the egg. Okay, the acrosome is responsible for the diffusion of the sperm to that of the egg membrane, which will be dealing in detail when we will read about the fertilization chapter. And what happened? The middle piece, the mid piece will be thickened and mitochondria will gather during this phase. As you can see in this diagram, this is the mid piece. In the Golgi phase, what will happen? The acrosome will be formed and the mitochondria will be formed in the mid piece, and the DNA will be condensed and attached in this phase. Condensed, the DNA will be present in the condensed manner. Okay. Next, what happens? DNA packing will be formed and then comes the acrosomal phase. Okay, acrosome will be formed and a cap-like structure will be formed. In this structure, you can see how the acrosome is formed. Is represented in this diagram. Golgi apparatus, acrosome vesicle will be formed, then spermatide nucleus, and gradually the growth will take place and finally the sperm will form. This is the process in which the sperm will be formed, motile sperm. Then comes the formation of tail, and the tail consists of temporary structure which only helps in the movement only. Okay, and it consists of machenty. Okay, and one more important thing, the tail will not enter into the egg membrane. Only the head and the middle piece will enter into the egg membrane. Then the maturation phase, that is the excess of cytoplasm will be removed and Sertoli cells will be present. Then spermation, that is the mature spermatozoa released by the protective Sertoli cell and into the lumen of the that is seminiferous tubule is called as spermation. And then takes place which removes the unnecessary cytoplasm. In this spermation phase, the basically what happens, the unnecessary cytoplasm will be removed. Okay, and here the total full organ of the sperm will be formed. That is the head, which contains the acrosome, nucleus. You can see this is a pink color cap-like structure, that is acrosome. Middle is the nucleus. This comprises of the head. Middle piece, centriole will be present. Centriole are of two types, distal centriole and proximal centriole. The proximal centriole will enter into the egg membrane. Distal centriole will remain. Okay, or sometimes both the centrioles also enter. This is the terminal disc and this is the cell membrane, and axial filament is responsible for holding the tail in position, and this is the tail, or you can say the flagella. This represents the active or the motile sperm, and now the sperm is ready to enter into the female genital tract for the fertilization process. And this is all about the process of gametogenesis. I know this is the only a very compact, or you can say a summary type, revision type. Many things are also there regarding detailed video also there, but I only want to give you a review, or you can say a summary regarding spermatogenesis, so that you can get a quick revision what is spermatogenesis and what are the important stages of spermatogenesis. Hope with this video you are clear with the first topic of spermatogenesis, and next in the upcoming part we will be discussing about, that is oogenesis, which is nothing but the formation of ovum. Okay, that's all for today. Have a nice day. Thank you.